Like I said, there are a lot of truncating proteins, but for some reason there's a bunch in this one that, once again, it's not a one-to-one thing. Even though this is low MSI, NMR is still okay, truncated proteins still occur there. I'm sure there's another, if you saw this, you could say, all right, well maybe I need to redo or do a different hypothesis and look at this instead of whatever I was looking at. Because for here, in this in particular, gene in particular, in APC, there's a bunch of truncated proteins there. In an area where if it's low MSI, where they have good NMR, we assume we shouldn't expect that, but we did. So this would be another area of research to look into going forward. I know it's boring. All right, so another one, melanoma, mutations, BRAF. Once again, what's the back from the case studies from before, which melanoma mutation is with the highest amount of melanoma? Which protein is it? BRAF, right? Isn't it BRAF? BRAF is the majority of people with melanoma have BRAF, but it could be other proteins as well. It could be the main driving mutation. But in this case, we're looking at BRAF. You can plug it in here, look at all the different ones. You can look at survival rates, all the patient info, gives us more information. Once again here, we have the BRAF. Usually on this slide, there's basically the other proteins that are there, the other genes, and then the percentages of those that are causing that led to that melanoma state. But the biggest one for melanoma will be BRAF. The majority of people with melanoma have BRAF mutation as opposed to other driving mutations. This one that's kind of funky. So look at the BRAF mutations. What they found is they've got red and blue. Red is, this is looking at survival rates. So once again, survival rates is patient info, only processed data. So they're looking here, red are people with BRAF, blue, people without BRAF. What does BRAF do, you guys remember? Let me ask you this, what happens when BRAF is, let's make it easier. When BRAF is mutated, what happens?
It's always on.
Yeah, you get uncontrolled proliferation. So if BRAF is mutated, you get way more proliferation. If I get way more proliferation, that's going to cause the cancer, right? Do I want that to happen? Is that good or bad? That's bad. However, why on this slide is it telling us if patients with the BRAF mutated melanoma, they have an increased survival compared to people that had other mutations that then led to melanoma.
If you have a lot of proliferation, then eventually it'll trigger apoptosis.
Exactly.
More than the other one.
Yeah, so both are still bad. People are still dying really, really high, right? It's not like it's kind of like a, I guess, glass half full kind of thing. But in this case, what we see is that if you have a high amount of BRAF, and this actually works for oncoproteins in general. BRAF is an oncoprotein. If you have a higher number of oncoprotein mutations within this cancer type, you have an increased survival. Because if you're proliferating so much, other parts of the cell are going to basically realize this, and they're going to go into not fight or flight, but they're going to go into the defense mechanisms where they're literally going to start saying, all right, let's make our checkpoints really strong and let's freeze the cell. Because we have way too high levels of oncoproteins. Too high level of oncoproteins means now those are too high, I then now increase the cell itself, will increase tumor suppressors, and it'll freeze the cell. And if I can't get rid of those oncoproteins, I go into apoptosis and the cell dies. So both these are deadly. They're both going to kill a lot of people. But in the case of if it's a high number of oncoproteins, in this case BRAF, if it's really, really high, the cell freezes itself. And then it goes through apoptosis, and then it'll die. That's not what cancer wants. So ideally, in that case with the BRAF, hopefully, not hopefully, but if I'm the cancer, the goal is to have other things get mutated, like other tumor suppressors or other checkpoint proteins. If I mutate those, then I don't arrest the cell, and the cell will continuously divide. But in this case, if it's just, in particular, a high number of oncoproteins, the cells identify that high number, and they freeze the cell, they freeze itself, and it kills itself to prevent itself from reproducing. That's what's going on here. And it's the same idea, looking at, this is not looking at BRAF, this is looking at all of the different ones that could be. Once again, high number of melanoma is due to BRAF. That's only 52%. The other percentages come from these other genes that can be mutated. That's the driving force in that case. If they look again, you look at only oncoproteins. Same thing as BRAF. BRAF is an oncoprotein. If you have, so on the exam, if he says, you're looking at melanoma, and there is a high level of BRAF or oncoprotein mutations, do these patients have an increased or decreased survival rate compared to other patients with melanoma due to non-oncoprotein mutations? The answer is a higher survival rate. because high oncoproteins, cell recognizes that, freezes the cell, apoptosis. So it's kind of counterintuitive to what we think about. All right, so now we're switching over from processed data to unprocessed data. Once again, big thing with unprocessed data, it's all the reads. It's literally all the DNA, and we're able to compare normal DNA versus other DNA. As in the patient, I can look at just their cancer cells versus their other cells. I couldn't do that with processed data. Processed data, it's already been done, and I can't look at that. So if in this case, I want to look at between a disease population and a normal population, I want to look at differences in in amino acid sequences. I need to use the unprocessed data. So what we do, so once again, just be aware of what we're doing here. It's the whole genome sequences, all of the genome. We have the processed, non-processed data for all of that stuff. We can still do RNA-seq. I'm assuming here the RNA-seq is from the transcriptome of everything. Disease patients, normal patients, versus the other transcriptome was just the disease patients, I'm guessing. So once again, how do we get it? We go through that Genome Database Commons, so just be aware of the GDC, Genome Database Commons, and tools like this, where literally you want to look at all the data for it. So once again, why do I want to look at unprocessed data? Let's use this as an example. And this is the big slide. So this is literally what you're going to get. And I blanked then wrote a code to then identify certain things in here. In the case, I don't know whatever this cancer is on the example from the study, what the actual disease state is, the actual gene isn't mutated. The gene is still the same. What's different is the regulatory elements. What are those again? That's the DNA outside of the gene. And what binds to the regulatory elements? Transactivators, right? And those then bind and then increase or decrease the chance of forming the PIC. So you guys already forgot it, it's okay. You guys know from med school though. So that's what this slide is showing us here. So now once again, there's no difference in the DNA. So if I went to see BioPortal and I just have the processed data, could I see this info at all in terms of issues with the regulatory elements? No, it's not in cBioPortal, because all it has is differences that we see of the allele of the actual alleles. We don't see that here. We're looking at not the gene itself. We're looking at the regulatory elements. So basically, what you have here, this is literally going down. This is an entire This is a regulatory element up here. So here is a C, here is a G, another G, so on and so forth. This is the actual sequences that we're going to see. And what we did is that basically here, look at the numbers. The first number is the number of times we see that in the normal population. The second number of times we see that in the disease population. And the third is the ratio between the two. So what happens and what they did is that they found people in the normal population. They had a certain number. So first of all, this should make sense. So at this first position, why do I have 0, 0, 0, but I only have numbers in one at this one down here? Why does that make sense? Not worry about the numbers within there. Why is it at position 1, there's only a C that's there, there's not any other thing that could have been there? It's a conserved sequence, because what is the same in everyone? We all have the same response elements. We all have the same DNA, right? That's the same everywhere. What's different between these two population groups is that in the normal people, they only had 49 of the C. They had the C 49 times. In the disease state, they had it, whatever it was, 129. So what we're seeing and what we go through as we move down, going across, we had a C here and a G here. As we go down and we see these differences that are occurring, and usually the biggest ones, the significant ones, this can be due to mutations or whatnot, what we see as we move down is that at every position, there is basically a threefold increase in the disease state versus the normal population. Normal population basically had the response element, but then in the disease state, they had those same response elements, but they were multiplied 3 times. So you had way more of the response elements. If I have more response elements, what does that mean? That means if I have transactivators, normally the limiting factor is usually transactivators bind to the response element. I can have a bunch of transactivators, but if I only have one response element, can multiple one of these bind to this? No. Because I've shown here, if I have a bunch of transactivators and I have 3 response elements, or 3 times as much, overall I get more stabilization of the PIC, and I make that gene, I transcribe that gene a lot more. That's what this is telling us here, is that there is a 3-fold increase in the response elements. That led to a 3-fold increase in transcription of that gene. And an increase of that gene, of that mRNA, means more protein, and therefore I get the disease state. So if you had to guess, what kind of protein would you guess that this probably is? An oncoprotein. I made way more of that oncoprotein, way more proliferation. What's that?
Just for that specific one, is that just like an arbitrary one he chose to point out? I mean,
Yeah.
The 3's coming over.
Yeah. He did this, and he wanted to show, like, you can still have some in the other ones too.
Yeah.
Once again, this is kind of like differences in population, but this is the significant one.
Okay.
And that's why normally it's like 3 for each of them. So the first number is normal, second number is in the disease patients, third number is just the ratio of the 2. So overall, we had and I know I wrote it like this for simplicity's sake, it's along the top, but it's not really exactly like that in terms of the orientation, but this is how you can think about it. There is a 3-fold increase in the overall number of response elements, therefore it led to more transcription. That's how I think about it. It's kind of confusing because you've got these here and these here for what's going on there, but once again, if you want to, it's not how it really is, you can think about it sort of like this, whereas in this case, it's repeated 1 time, but in the disease states, that same thing is repeated basically 3 times in a row or whatever. So therefore, more transactivators combined and led to increase in that transactivation. So once again, other thing, you can look at blood samples. You can look at one patient in particular, and you can compare the blood versus the I think it's the colon cancer. I think so. Let's just say it's colon. Some cancer type and blood. Once again, in process data, the only thing it always shows you is the gene of the tumor cell versus the sequence gene of that same gene in the overall in that same person, but the normal healthy cells. You're always comparing the screwed up DNA of the cancer cell line versus that same DNA everywhere else in that person. So, if you guys remember FGF receptor mutations, what happens if you're born with the FGF receptor mutation? You're going to have what disease? Dwarfism, right? Would that show up, I know it's not cancer, but would that show up in process data? Why not? Because all of my cells have what? That mutation. They all have the same sequence. There's no difference between the 2. Will that show up in process data? No. Why? Because you had it from birth. So that's the big thing here. If we have mutations due to mutations that we had during gamete formation or during fertilization, that doesn't show up in process data. Why? Because process data is always comparing the tumor gene versus the other gene, all of the same gene in you, but in all your healthy cells. So the mutation had to occur later in life. If, or in this cancer type, we were born with it, so it basically was, but it only affects, remember, the same mutation affects different cells differently. In this case, majority of your organs are fine, whatever one this is, with that mutation leads to that increased proliferation and cancer. So in this case, that's why we can look at the blood versus, we can look at blood versus their normal, the actual cancer cell type, and even if both those are the same, we can still see that data if it's unprocessed. We can still see, okay, well, there's no difference between the 2. However, now I can compare that gene, this sequence, in this disease state versus all the other healthy people. And in that case, I will see a difference, right? Because they had the mutation from birth. So once again, the big thing is knowing what process data is always comparing. disease allele, the disease gene, versus the healthy gene in that same person, or the other genes in all the other cell types. If it was a disease like this, it would not show up in process data, because even the blood has the disease, but we weren't able to identify it. We compared them and we saw no difference. So therefore, for us, this is not causing the cancer, even though it actually is. So the idea is like big data, looking at obviously given topics, looking at massive amounts of data and comparing things like all those transactivators, or to all the response elements, things like those massive the mutations that occur in all the different cell types going from there. It's much worse looking at clinical information. Once again, when we see clinical information, think process data. See BioPortal. We're able to see all of these mutations led to this state. Therefore, we see these sort of symptoms, these outcome potentials, things like that. Last one here, another one by Blank. This one's pretty obvious. Basically, if you're a smoker, you have more lung mutations in your lungs. Hopefully that makes sense. And obviously, if you stop smoking, mutation rates go down. That also makes sense because if you stop smoking, you may still have lung cancer, but a lot of those cells will go through apoptosis on their own. Keep in mind, your lungs are able to make more. You have basically a somewhat type stem cells that are able to make more of your alveolar cells, those cells you need for that as you go forward. Once you get a really low p-value, that's what we want to see. So yeah, that one's pretty straightforward. Looking at that. And once again, where we want to go in the future is just looking at other ways we can use the unprocessed data. That's it's a gold mine things to use, things that we want to be comfortable with. And then last thing I want to talk about is just the idea that we've only talked on cancer for this lecture. There are other databases as well that do a similar thing where there's processed data. Some of those like Alzheimer's genome analysis. The problem with that is that you basically have patients have to be deceased. You can't kind of go in and take out their brain and look at the DNA there after they're gone, unfortunately. But the goal is maybe find other browsers for other kinds of diseases, and therefore use those, compare those to the normal genome browser, and see what we can find from there. So that's the next step, is taking it from cancer to other, I'm sure there's already been research in there, so taking it into other areas. And yeah, that's that one. Like I said, I'll be here for a while. If you have any questions, feel free to ask me. If you want to, keep in mind that Sunday before, after Thanksgiving, Sunday before anatomy, I'm going to do the cancer one. And that'll just be, it's like 80 slides. It's a lot of stuff. And I gave you those slides, I didn't give you those. Make sure you read over that stuff, though, because whatever I think is important, many people still want to focus on. And I'll do that. And then we'll have one last review on the Friday before. After anatomy, we'll have my last review. That'll be for the last lecture on personalized medicine. And then any questions you all have. Yeah.